Hello, everyone, and welcome back to Science in the Age of COVID-19. Today we have a very special guest, Jason McLellan, a professor of molecular biosciences at the University of Texas. Jason is a structural biologist working mostly, but not exclusively, on viral structures, mostly, again, but not exclusively, on their capsid proteins. Determination of these structures is critical. It lets you know what you're dealing with, helps you understand the basic biology of what receptors and self-surface proteins they're recognizing, how they're fusing to get into the cell, helps you design drugs, helps you evaluate vaccine candidates, really critical stuff. And really driving home the important how important this is. Jason's group was the first one to publish the Cryo-Em structure of the prefusion complex of SARS-CoV-2 in 2020, and I have never seen anything like this. The paper has been cited more than 4,300 times in less than a year, which is just truly remarkable. So yeah, that really enabled all the subsequent work on things involving the structure of the viral capsid. And today he will tell us about the spectacular work on next-gen vaccine design. So Jason, take it away.
Well, thank you for the introduction. Yeah, it's been kind of amazing that it's been cited over 4,000 times. So that will be my highest cited paper. So it's kind of all downhill from here. But there's a lot of interest in some of our work on the vaccine development for SARS-CoV-2. So I thought I'd tell a little story dating back to my time as a postdoctoral fellow working on a different virus that is kind of the love of my life for virus, respiratory syncytial virus. Kind of give an update on that and how we use a lot of that same information in our work on coronaviruses. All right, so I do have a lot of conflicts of interest and I'm obligated to share. So I am an inventor on patents related to prefusion RSV-F and its use on prefusion stabilized coronavirus spikes and their use, and other patents on monoclonal antibodies and camelid antibodies for RSV and coronaviruses. All right, so I mentioned the first part will be on pneumoviruses. And a lot of my work when I was a postdoc at the Vaccine Research Center, and then second or most of the part on coronaviruses, work that I did at Dartmouth College, where I started my lab, and then more recently here at UT. So two things that these viruses have in common are that they contain class 1 viral fusion glycoproteins. And class 1 viral fusion glycoproteins, some of the more common ones are shown here. So coronavirus, the spike protein, that's the largest class 1 viral fusion protein. Also HIV envelope, which gets called to GP120, GP41, influenza hemagglutinin, and then the F proteins from paramyxoviruses and pneumoviruses. So these are all synthesized as single chain inactive precursors, and they need to be proteolytically activated at one critical site. Some of these proteins have two cleavage sites. The RSV-F protein contains two cleavage sites. Coronaviruses contain two cleavage sites, but one of them is strictly required. And although the cleavage site at the S1, S2 junction in coronaviruses, and particularly SARS-CoV-2, where it's this insertion of a furin site, gets a lot of attention, it's actually the second cleavage site in the spike protein, which is called S2 prime. That's the one that's strictly required. And so what you can see is that something like the coronavirus spike, it contains two subunits, and the S1 subunit is what you can think of as a fusion suppressive cap. Its job is to prevent premature triggering of the S2 subunit, which is all the fusion machinery. Right? So the fusion machinery is a hydrophobic fusion peptide, a transmembrane domain, and then two heptad repeats that will interact with each other to form a coiled coil. And because these are all trimeric proteins, you end up with a trimer of coiled coils, which is a 6-helix bundle in the post-fusion conformation. So the coronavirus spike, the S1, that binds to protein receptors, and it can also has another domain, the N-terminal domain, that can sometimes bind to glycans on the cell surface. We can see that HIV-1 envelope, the GP120, the fusion suppressive cap, is much smaller, but this still binds protein receptors like CD4, CCR5. Influenza HA1 is even smaller. It doesn't bind protein receptors, it binds just sialic acid. And then you can see that the F proteins for like RSV, paramyxoviruses, those have completely lost the fusion suppressive cap. They're essentially just the unstable fusion protein. How those trigger to refold at the proper time and place is still a mystery. Whereas coronavirus and HIV-1, they use receptor binding to cause triggering of the glycoprotein, and influenza uses low pH. We still really don't understand how F proteins refold at the proper time and place. And maybe they don't, maybe they just spontaneously refold at some rate. So RSV and also metapneumovirus, which is another one we work on. These are members of the pneumovirinae family. It's more recent. they used to be paramyxoviruses that were reclassified into their own family of pneumoviruses. These bud as filaments. This is really beautiful Cryo-Em from Elizabeth Wright's groups. They have a mammalian cell grown on the EM grid. You can see these RSV filaments budding. On the surface of the RSV variant, there's two proteins: the F protein, which I'll be talking about, and then the attachment protein, the G glycoprotein. So this is how we think the F protein works. It exists in an initial, what we call metastable, kind of unstable, pre-fusion conformation. As we mentioned, it either spontaneously triggers or there's some unknown triggering event that causes it to go from the pre-fusion to the pre-hairpin intermediate. This is where it throws the hydrophobic fusion peptide into the host cell membrane. This then begins to collapse down to form the post-fusion, highly stable conformation. That difference in free energy between the metastable pre-fusion state and the highly stable post-fusion state provides the energy to fuse the two membranes together. When we started this work back in like 2009, when I was a postdoc, our goal was to try and create an RSV vaccine. There had been a lot of failures, including a really disastrous clinical trial in the 1960s that resulted in 80% of the vaccinees being hospitalized upon natural infection and 2 of the infants dying. And so that's really slowed down RSV vaccine development. And we wanted to try and come up with a structure-based design of like the optimal antigen. And we wanted to focus on the F protein because it's strictly required for entry. The G protein can be deleted and you still get infectious virus, but you have to have F. So when we started this work, we didn't have much in the way of high resolution information. We had some rough secondary structure. Several neutralizing antibodies had been isolated by people in the field. They were targeted against these antigenic sites called Site 4, Site 1, Site 2. Synagis is a monoclonal antibody. It's also called palivizumab, developed by MedImmune, which is now AstraZeneca. That antibody targets antigenic Site 2, and it is used in the clinic for high-risk infants, premature infants, infants with bronchopulmonary dysplasia. It can be given to them passively each month during the RSV season to reduce severe infections. And so we at least knew that where this antibody existed, it provided a rationale that if we could elicit similar types of neutralizing antibodies from a vaccine, that we could maybe protect against severe RSV disease. So to get the first structures of the RSV F protein, I tried making just an ectodomain. We just deleted the transmembrane domain. That resulted in a lot of aggregation. And I looked back and saw a nice paper from Jose Malero's group in 2004, where they showed that they also just deleted the transmembrane domain. They got these rosettes, kind of like spoked wheels. And they hypothesized that it's post-fusion molecules, each spoke, being held together by the hydrophobic fusion peptide. And so if they deleted the first 9 amino acids of the fusion peptide, Then they got just the individual spokes, and it was no longer these rosettes. And so I made that and was able to crystallize it and determine the structure. This was published in 2011 within a week of a group from Novartis Vaccines, Kurt Swanson and Phil Dormitzer. And so this revealed the first structure of the RSV protein and the post-fusion conformation. What was interesting is that we saw the known antigenic sites, sites 2, site 1, site 4, those were all present and surface exposed. And the neutralizing antibodies, so that targeted site 4, Synagis, the one used in the clinic, those could bind post-fusion, suggesting that this is actually a decent vaccine antigen that could elicit at least site 4 and site 2 antibodies. So Barney Graham's lab, they immunized mice with either RSV or a formalin inactivated RSV or the purified post-fusion F, the crystallized. And it was able to elicit some neutralization titers, EC50s in the hundreds, so it wasn't bad. And this formed the basis for a number of vaccines that were moving through the clinic. So this is a snapshot from the organization PATH. They update this every so often. So this was from 2016, just showing, one, there's a lot of activity for RSV vaccine development because it's really the last major childhood pathogen for which we don't have a vaccine. You can see the different modalities. There's live attenuated. It's primarily being evaluated in seronegative infants. Then you have more particle-based subunit, which is like purified protein, some of the nucleic acid, Inovio, gene-based, Moderna, and others. And so the ones that were in 2016 furthest along in clinical development were some post-fusion F proteins from GSK, from MedImmune, and then Novavax has what they call the nanoparticle, but it's actually just these rosettes. But nanoparticle sounds better. And unfortunately, all these post-fusion based vaccines failed in their phase 3 clinical trial. So Novavax has failed their phase 3 in the elderly. MedImmune's post-fusion F vaccine failed their 2B, also in the elderly. That was a purified protein that was adjuvanted with a toll-like receptor 4 agonist. Actually, maybe had negative vaccine efficacy, so that was suboptimal. And then again, Novavax's vaccine failed. This was 2019, so 2 years ago. I think this was in pregnant women. So it was pretty clear that post-fusion F is not a great vaccine antigen, and that we needed something else. And obviously, we probably want to use prefusion. That's the active form of the molecule. That's the molecule that exists primarily on the surface of the virion. The problem is that it's so unstable. Every time you try and purify F, you get post-F. You can't lock it in pre-F. So our idea was, can we come up with a way of locking the F protein in the prefusion conformation, first initially to get a structure, and then to make some structure-based engineering changes to actually lock it as a prefusion F molecule for use as a vaccine antigen. So in 2011, Jose Malera's group identified that humans, upon natural infection, actually make a lot of antibodies that are neutralizing, very potently neutralizing, but they don't bind post-fusion F, suggesting that they're specific for the prefusion molecule. So Barney and I were thinking that if we could get prefusion F-specific antibodies, we could co-express F in the presence of these antibodies. If F first transiently folds into prefusion before triggering the post, then these antibodies could bind, lock F in the prefusion conformation, and it could allow us to get the structure. And so this was a couple of years of work, but we were able to get some prefusion F-specific monoclonal antibodies. One, 5C4, came from a collaboration we started with a group in Xiamen, China. They were able to immunize mice with an adenovirus vector encoding wild-type F. They screened several thousand hybridomas. They tested them all for neutralization activity. So more potent are to the left. So Synagis, which is the one still used in clinically, is shown here. And so this one antibody that isolated 5C4 was 2 logs more potent. And then most excitingly, it didn't bind to post-fusion F based on an ELISA. So since we could make post-fusion F protein, we could coat. So Synagis in gray binds post, as we knew, but 5C4 doesn't. And then we were able to obtain 2 additional antibodies, D25 and AM22, from a company called AIM Therapeutics. These were isolated from infected humans. These also had the same properties, extremely potent neutralization and lack of binding to post-fusion F. So then I was able to get this approach to work where in just one transient transfection, we could transfect in 3 different plasmids, one encoding F, and one encoding the heavy chain, and one encoding the light chain, and out would be secreted a complex. And we could do that for all 3 of them. And I was able to get crystals and eventually solve the structure to about 3.6 angstroms of D25 bound to RSVF in the pre-fusion conformation. And we could also see from negative stain EM that AM22 and 5C4 also all bound this epitope at the apex of the pre-fusion F molecule, which we called antigenic site 0. And so we now had the post-fusion and pre-fusion structures. It revealed a lot of interesting biology, that the fusion peptide, which is hydrophobic stretch of amino acids, that's buried inside the trimer, sequestered away from the solvent. The F1 and F2 subunits aren't like 2 separate entities. They're just completely intertwined. This is essentially the coronavirus S2 subunit. Looks very similar to this. We could see, if we look at an individual protomer, about half of the F protein is in the same conformation between the pre-fusion and post-fusion. We can see this helix loop helix region. This is the antigenic site 2. This is where talivizumab binds. So it is able to bind post-fusion, but it neutralizes because it binds pre-fusion as well. And so we could see that the N and C termini of the F protein undergo really dramatic conformational changes. So the C terminus of F1 ends up flipping all the way around to form the outer helix of the 6 helix bundle. And the N terminus of F1 is a really dramatic refolding and rearrangement. So you have the fusion peptide, then a helix, turn helix, a beta hairpin into this alpha 4 blue helix. All of this flips and refolds. All the coil, the hairpin refolds into one long helix in the post-fusion conformation. So with those structures, then we could start designing substitutions that would lock the protein in the pre-fusion conformation. So that way we could express and purify pre-fusion F in the absence of the D25 antibody and use it as an immunogen. And so we tried a couple of different strategies. I designed a disulfide bond. One cysteine was introduced in a region in blue that undergoes a conformational change between pre and post. Another cysteine was in a region in gray that doesn't undergo the conformational change. This was really nice because there's actually 2 serines that normally hydrogen bond. So changing them to cysteine is pretty straightforward, and it forms a disulfide bond. And we also introduced in, we call cavity filling substitutions. These proteins are not really well packed and have little pockets and cavities. So that way they are unstable and can undergo the conformational change. And so we could fill some of the pockets, like mutating this serine to a larger phenylalanine, fill the pocket, made it entropically unfavorable to expose that phenylalanine to solvent. And so this molecule we could express and purify, and it stayed in the pre-fusion conformation. It bound D25. And when Barney's lab tested this in both mice and rhesus macaques, we were able to get really high neutralizing antibody titers. And so immunizing with post-fusion, again, we would get EC50s kind of in the hundreds, but with the pre-fusion F molecule, we were 1 to 2 logs higher neutralizing antibody titers. And these were the highest titers Barney had ever seen in his career. And so we were really excited by this. And this was a runner-up for the Breakthrough of the Year in 2013 in Science Magazine for proof of principle for structure-based vaccine design. And it's continued to move through clinical development at the normal pace of vaccine development, unlike the rapid coronavirus pace. So we created that molecule in 2013. In 2019, the interim results from the phase 1 were made available. So these were healthy adults, 18 to 50 years old, receiving 2 injections 12 weeks apart, receiving either 50 150, or 500 micrograms with and without alum. So these are the results after just the first dose. And particularly in like 150 microgram, we saw a really nice increase in neutralizing antibody titers. Again, well, I haven't mentioned, but all adults have been infected by RSV probably multiple times. By the age of 2, we're essentially all infected with RSV. So we're not immunizing a naive population like we are for SARS-CoV-2. We're essentially boosting a seropositive population, but we're able to obtain somewhere 10 to 15-fold increases in neutralizing antibody titers, which have maintained at that level now. There's some follow-up data was just published last month that even like after a year, we're still relatively high. And so that's really exciting. And so Prefusion F has really now taken over the field for RSV vaccine development. GSK is pushing our molecule, DSCAV1. They've just started phase 3. Pfizer has their own, which is essentially my molecule, but with some different substitutions to maybe get around our IP, and we'll see how that goes. Companies like Icosavax and others have self-assembling nanoparticles displaying our DSCAV1 molecule. Then you have some of the nucleic acid, adenovirus from Janssen, Moderna working on that. So it's really exciting. And at the end of last year it was when GSK and Pfizer both started their phase 3. So we're really optimistic based on some of the phase 1 and phase 2 data that this could result in the first vaccines for respiratory syncytial virus. All right, so that's been going on since 2009 or so. And that brings us to coronaviruses. So as you know, the coronaviruses are covered in the spike protein, and that's related to the RSVF protein. That's the spike gave the virus its name. It's from a nice Nature little article in 1968, how the appearance of the virus, which has this characteristic fringe of projections, recalls the solar corona. So they put forward the name coronavirus. And so now, based on a lot of structural biology from our group, David Diesler's group, Andrew Ward's group, we have a pretty good understanding of how these spike proteins work. They first are involved in attachment to the cell surface. In this case, for SARS-CoV-2, it binds to the ACE2 protein. It has receptor binding domains that can move up and down to hide and expose the ACE2 binding site. Upon ACE2 binding, we think all 3 of the RBDs eventually get trapped up, which is unfavorable. And then we get shedding of the S1 subunits, kind of breaking away from the prefusion S2. That then undergoes a conformational change, like we showed for RSV, where the fusion peptide is thrust into the host cell membrane. This then begins to collapse down as the coiled coils find each other and eventually form the highly stable post-fusion conformation with the 6 helix bundle that then fuses the viral membrane and host cell membrane, forming the fusion pore that allows the genome and other contents of the virus to enter the cell. So I left the VRC in 2013 to start my own lab at the Geisel School of Medicine at Dartmouth. And that was around the time when the Middle East respiratory syndrome coronavirus had emerged in Saudi Arabia and the surrounding region. And Barney and I thought that this would be a great test case to try to apply everything we just learned for RSV and structure-based vaccine design to a new family of viruses, coronaviruses. At the time, we didn't have a lot of structural information on the coronavirus spikes. All we had were crystal structures of some individual domains from Fong Ling and others. So there were structures of the N-terminal domain of the S1 subunit, which showed a galectin-like fold that can bind to various sugars. There is multiple structures of the C-terminal domain of S1, which is most often referred to as the receptor binding domain, or RBD. That revealed this beta sheet, a conserved beta sheet that then has these different extensions coming off that are the receptor binding motifs. For S2, there wasn't very much. It was just the coiled coil, the 6 helix bundles of the 2 heptad repeats in the post-fusion conformation. So we really had no idea how this trimeric molecule assembled in the pre-fusion state. Fong Li and Stephen Harrison had some negative stain EM of full trimers from SARS-1 from 2002, showing these like 3 lobes. There's some additional kind of lower res EM. So even by 2016, this is mostly what we kind of thought the spikes looked like. And so working with Barney Graham, we started trying to make the MERS coronavirus spike ectodomain for structural studies, and we just could not make a lot of it. We could make some of the individual domains, we could make the N-terminal domain, we could make some of the S1, but when we tried to make the full spike ectodomain, we just couldn't produce it. We did have an antibody available that recognized the MERS-CoV spike. We could pluck out some complex that looked okay, but a lot of it was really like a heterogeneous mess. Fortunately, Barney's lab was also working on HKU1, which is one of the 4 seasonal coronaviruses, along with OC43 and the 2 alpha coronaviruses. And the HKU1 spike, for whatever reason, it was just a lot more stable. And when we expressed it, it looked really good. So we can make a lot of the spike ectodomain. If we kept a wild type cleavage site in between S1 and S2, we could get nice cleavage and we could see the 2 subunits. We could mutate out the S1, S2 cleavage site and just get the single like S0, that looked good. And our favorite construct had the S1, S2 cleavage site mutated with also this fold-on trimerization motif appended to the C terminus, trying to replace the transmembrane domain. And this molecule looked really good. And we sent it to Andrew Ward's lab, who's an expert in electron microscopy. My lab was not doing Cryo-Em at that time. So we were working with Andrew and their group with Rob Kirchhofer, Jesper Paulson, others, were able to get the first structure of a human coronavirus spike protein. And it revealed this really intricate arrangement, the 3 protomers. So this is looking down towards the viral membrane, white, gray, and ribbon. We can see the N-terminal domains are located at the periphery. The trimerization is all mediated by the receptor binding domain. This also identified 2 new subdomains that we were unappreciated or unidentified before, what we call subdomain 1 and subdomain 2. And then most importantly, we could see the arrangement of the S2 subunit and its pre-fusion conformation. The fusion peptide is actually localized to the outside rather than buried in the inside, like we saw for RSVF. And this explained a lot of biology. It was also really useful for our structure-based vaccine design efforts. And I think you can see a lot of similarity between the coronavirus pre-fusion S2 subunit and the RSVF protein. So they both have a central helix shown in blue. Everything's just a little bit longer and larger in coronavirus. So here's the central helix in blue for the S2. Here's a smaller central helix for RSVF. Then we can see that the first heptad repeat is in green, yellow, orange, red, and then has this magenta region containing the fusion peptide. That's similar where we have green, orange, yellow, red, and then this purple region containing the fusion peptide. And so all of this just flips like 180 degrees and begins polymerizing into the really long helical bundle in the post-fusion conformation. So this allowed us to use some of our prior knowledge on stabilizing RSVF to also stabilize coronavirus spike proteins. And one of the tricks we used was the introduction of proline substitutions. We tried other things like disulfide bonds, but what we were doing is that we had the structure of HKU1 and we were trying to engineer MERS. And those just aren't close enough. And to introduce disulfide bonds, you have to be within an angstrom or 2 to get it right. And the structures are just too different. But prolines had been used dating back to 1998. This is from Judy White's lab, which is really beautiful work. HA doesn't need to be stabilized in the prefusion conformation because at pH 7, it is in the prefusion. But they were able to introduce prolines into this region between the central helix, shown in orange, and the first helix of the heptad repeat 1. And they introduced 2 proline substitutions. And when exposed to low pH, it resisted the conformational change. John Moore and Richard Sanders introduced a similar proline substitution in HIV-1 envelope, which is the basis of this BG505 SOSIP trimer a lot of groups have used. And then our colleague at Janssen, subsidiary of Johnson & Johnson, Johannes Langendijk, he came up with his own way of stabilizing the RSVF protein in the prefusion state by also introducing this proline substitution between the central helix and the first heptad repeat. And so my postdoc, Nian-Shuang Wang, started undergoing this proline scanning idea in the hinge region between the central helix and the first helix of the heptad repeat. And you can see that when we tried to make the wild type MERS ectodomain, it just got very little protein. But individual substitutions of these residues to proline resulted in really large increases in protein expression. And the one that we liked the most had two adjacent residues substituted to proline, and we got a really huge increase in expression, close to 50-fold. And what's exciting is that this region is really well conserved at a sequence level and structural level between all these different beta coronaviruses from the different lineages of beta COVID. So HKU1, MERS, SARS-1, they all contain this RLD, conserved glutamate, and then this first part of the central helix is all conserved. And so the two residues we substituted were at these positions, and this resulted in this really highly stable spike protein for MERS. So you can see based on size exclusion chromatography, the wild type spike protein, which we could barely make, had this tiny little peak. And when Andrew's lab looked at it by negative stain EM, it was this heterogeneous mixture of post-fusion and pre-fusion spikes. But with just these two prolines substituted in to this 1200 amino acid protein, we got this really large boost in expression, and it was highly stable. And when Andrew's lab looked at it by negative stain EM, it was just all homogeneous pre-fusion spikes. And Kazmekia Corbett in Barney's lab, she immunized mice with either the two proline stabilized MERS spike, wild type spike, S1 subunit. And what we could see is that particularly at the lowest dose, the 0.1 microgram dose per mouse, we could start to see differences, statistically significant differences, showing that the two proline form of the spike was more immunogenic than either the wild type or just the S1 subunit. And because that region is so well conserved, we could just by sequence alignment, introduce those two prolines into like SARS spike. The SARS wild type spike, again, this is SARS-1 from 2002. We could make a bit more of it, so that peak was a little bit higher than for MERS. Both the two proline changes, we got a nice boost in expression. SARS wild type was also this mixture of post-fusion and pre-fusion spikes, but with the two proline substitution, it was all homogeneous pre-fusion spikes. And it worked for HKU1, which is a lineage A beta coronavirus. The wild type was already pretty stable, and we were able to get a structure from it, but the two prolines boosted expression. It also worked for OC43. Even a SARS-like back coronavirus that had yet to emerge into the population, that also benefited from the two prolines. And so by 2017, 2016, we kind of had this universal method of stabilizing beta coronavirus spikes by substituting those two residues with proline. So that allowed us to move really quickly. At the beginning of last year, by this point, my lab had moved to the University of Texas at Austin because we wanted to have our own cryo-electron microscopes and doing Cryo-Em. And so on January 10th, the novel coronavirus genome sequence was released. Within 10 days, my postdoc, Nian-Shuang Wang, had cloned the DNA containing the two proline spikes. 10 days later, my graduate student, Daniel Rapp, had purified the spike from mammalian cells, froze grids, and started Cryo-Em data collection all on the same day. And then 12 or so days after that, on February 10th, we had determined the structure, submitted it to Science and bioRxiv. So about a month from novel coronavirus genome sequence being released to a submitted manuscript describing the spike structure. So our construct for the SARS-CoV-2 ectodomain was just up to residue 1208. The proline substitutions were at positions 986 and 987, which correspond to these regions. We also mutated the S1, S2 furin cleavage site and replace that polybasic region with glycere, allicere. And we could make a little bit of the protein. It wasn't great. It wasn't the robust expression that we had seen from MERS and SARS. We were only able to get about 0.5 mg per liter, but it was really well behaved. And as I mentioned, Daniel was able on that same night to collect a Cryo-Em data set that went to about 3.5 angstroms with no symmetry and 3.2 angstroms if we applied C3 symmetry. And yeah, here's the structure of the SARS-CoV-2 spike protein. What was different from what we saw with the MERS Cryo-Em structures was that all the particles had one RBD in what we call the up conformation. For MERS, there's actually a mixture of like having all three RBDs down. Some particles had one RBD up, some had two RBD up. Here, for this construct, under these conditions, almost all the particles had one RBD up, accessible for ACE2 binding, but the other RBDs in white and gray were in the down conformation. We could see a lot of interesting motion in our data set. So we did a 3D variability analysis of all the individual spikes that went into the reconstruction. We can see that there's a range of motions in the RBD, sampling more down and more up conformation. When the RBD moves up, you get this concerted motion of the neighboring RBD and NTD moving in to fill that void. And so these are really dynamic molecules, and we're still trying to understand all the different conformational changes, particularly at 37 degrees. I mean, these are ectodomains frozen in vitreous ice. And so we're really keen to see how this looks at 37 degrees on the surface of a virion and understand the conformational changes between 3 down, 1 up, 2 up. But with structures from my group, Wiesler group, Bjorkman group, some really beautiful cryo-ET from John Briggs and others, we now have a really great understanding of what SARS-CoV-2 virion looks like. And this is an animation, but it's based on tons of structural data that there's 25 to 35 spikes. The RBDs are in 3 down, 1 up, 2 up conformations. There's a long flexible stalk that's usually disordered in the Cryo-Em single particle studies that can be observed by cryo-ET, and it's flexible. It allows the spikes to move. I think I saw that there's been over 1,000 structures for SARS-CoV-2 deposited in the last year. So it's great to see the structural biology community coming together to learn a lot about this pathogen. What we're excited about as a lab and as part of our collaboration with Barney and Andrew is the adoption of the 2 proline spike in it's now 5 of the leading COVID-19 vaccines. So Moderna's mRNA 1273, that encodes for the full spike containing the 2 P mutations at 986 and 987. Pfizer BioNTech, they had 2 mRNA vaccines that they were testing. One was just RBD, and then the other was this full spike containing the 2 prolines at 986 and 987. That's the BNT162b2. That's the one that's being used in the US. That's what I was immunized with. Johnson & Johnson, they tested 7 different spike inserts and flavors in a really nice paper of Dan Baruch. They have the Ad26 vaccine. From their data, they showed that the 2 proline substitutions and mutated furin site gave the best immune responses in their preclinical work in the macaques. And the Novavax, we're still waiting on data from. They have a full-length detergent purified subunit vaccine that contains mutated furin site and also the 2 proline substitutions at 986 and 987. And CureVax also contains that. So it's been really exciting to see something that we designed back in the lab actually being given and used in humans. So it kind of leapfrogged RSVF, which we thought would maybe be the first vaccine that includes structure-based vaccine design elements. And now these coronavirus vaccines are. But we weren't happy with just the 2 proline spike. We shipped that plasmid out to over 100 labs around the world, and people could not make the spike. And they're having a lot of trouble. So Bill and Melinda Gates Foundation reached out to us to see if we could make a better spike. Now that we had the structure of the actual SARS-CoV-2 spike, we could perform a second round structure-based vaccine design. And so, sure, let's try and do it. We can probably do better. Our goal was to improve stability and increase the expression about tenfold, get it closer to what we had with our MERS and SARS-1 spikes. So my postdoc, Qinglin, and I, we designed about 100 different substitutions based on the SARS-CoV-2 Cryo-Em structure. All the substitutions were in the S2 subunit and kind of grouped into 4 different categories. Some were the introduction of prolines, because if 2 is good, maybe more is better. Those prolines were designed to cap alpha helices and rigidify some flexible loops at protomer interfaces. We also introduced salt bridges by mutating or substituting a residue to a charged residue. So in this case, this is a really nice one. It's normally a threonine. We mutate it to an aspartate. It can then form a salt bridge with an arginine across the neighboring protomer. Cavity filling substitutions, mutating like leucine to phenylalanine to better fill some of these pockets. And then the introduction of disulfide bonds form these covalent staples between regions that move and don't move between the pre and post fusion conformation. So this was work that was performed with 2 other groups. So after Qinglin and I designed all these, we now had to like test 100 different spikes. So 2 groups at UT Austin, Jennifer Maynard and Ilya Finkelstein, they donated their students and postdocs around like April of last year to just help clone, express, and test all these. So 100 different variant spikes were cloned, expressed, purified, and characterized. So here we're looking at the expression levels. These are all from the same culture, like 40 mil cultures. So S2P, we were getting around a microgram per mil, 1 mg per liter. The salt bridges, many of them look really quite good. We're getting 3 6-fold increases, 2-fold increases with some of those. The prolines, also with a number of prolines, this A942P was really impressive. Also this one near the fusion peptide, even 50% increases, 2-fold increases. We like those because they can be additive or sometimes even synergistic. Disulfides sort of underperformed. Some of them helped increase the melting temperature. Didn't really boost expression very much. Cavity filling and some of the ones where we introduced hydrogen bonds, those also looked pretty good. So all of these were purified by affinity columns. Anything that didn't kill expression, we then put over size exclusion columns. We could see the nice boost in expression. So the dashed black line is the S2P. Disulfides looked okay. This one boosted expression because of the leftward shift, which it's still a well-folded spike. We think its radius of gyration has increased either because more RBDs are in the up conformation or because the trimer splits open transiently into like 3 protomers that then reassemble. I think it's probably the latter now. We have some additional data. But the proline variants also look really good, see nice boost in expression compared to S2P, and we're running at a similar retention volume. So with these 25 individual beneficial substitutions, we then had to come up with methods for combining them all. So we started trying like, let's put all the salt bridges together. Let's put all the prolines together, all the cavity filling. And then also like mix and match, take the best from each one and make like Frankenstein monsters. So just by putting the prolines together, we kind of hit our initial goal of boosting expression tenfold and increasing stability. So we kind of just stopped and wanted to get the paper out and the plasmid out and get it to people working on this stuff. So we can see that the S2P expression shown here, these combo 14 and 45 contained 2 additional prolines on top of the original 2, and then combo 47 had 4 additional prolines. Combo 47 we liked a lot, so we're seeing a nice tenfold increase in expression compared to S2P. Based on differential scanning calorimetry, we saw about a 5-degree increase in melting temperature for combo 47 compared to S2P. And by negative stain, it just looked great. It was just all homogeneous pre-fusion spikes. So since it contains 6 total prolines, we called it Hexapro. At large scale, it's been great. We normally get 11, 15 mg per liter, all pre-fusion spikes. That's from Freestyle 293 cells. If we switch to the XP cells from Invitrogen, we're getting 30, 40 mg per liter, and still just a lot more than S2P. One group got 130 mg per liter with a prototype cell line that Invitrogen's working on. We tested binding to ACE2. It's basically the same between S2P and Hexapro, 11 versus 13 nanomolar, which is within the error of the experiment. We didn't expect any ACE2 affinity to be different. All the prolines are in the S2 subunit, far away from the receptor binding domain. We also qualitatively assess stability. So we were just looking by negative stain EM. So this is looking at freeze thaws. So both the 2-proline and Hexapro can survive 1 freeze thaw. After 3 freeze thaws, the 2-proline form is basically gone. There's almost no prefusion spikes. It's all aggregating and falling apart. About 80% of the particles for Hexapro are still all prefusion spikes. We could also let Hexapro sit at room temperature for 1 day, 2 day, looked fine. Or S2P, we started seeing it kind of break down. We could also stress test Hexapro, heating up to 55 degrees, even 60 degrees for 30 minutes looked okay. Whereas S2P by around 50 degrees started breaking down again. We wanted to make sure that the introduction of the 4 additional prolines did not substantially alter the structure of the spike protein. So Daniel in my lab determined a Cryo-Em structure of Hexapro. It's about 3.2 angstroms with no symmetry applied, I think around 2.9 or 3 with C3 symmetry. So in white is the S2P Cryo-Em structure, in green is the Hexapro structure. So you can see all throughout S2 where the prolines are introduced, it's just the same, superimposable. The density was really nice in S2, so we could see that the introduced prolines were in the conformations that we had designed them to be in, which was satisfying. So globally, even locally, there's really no changes in the conformation of the spike protein. We wanted to make sure we didn't alter the antigenicity. So we worked with Greg Ippolito and Jason Levinder here at UT. So this was like middle of last year. They had 4 samples of patient sera who had been infected with COVID-19, 117, M2, H2, and C2. So they performed ELISAs with plates coated with either Hexapro or S2P. And so for each of the patient sera in the different colors, you can see there's essentially no difference between Hexapro and S2P, which is good. So we haven't altered reactivity with antibodies. This is unpublished data that I hope gets published soon by Barney. We sent them the 2-proline and the 6-proline spikes for immunogenicity experiments. And so Olu Abiona and Kosmetki Corbett, they immunized BALB/c mice 10 per group at week 0 and week 3, and then performed a terminal bleed at week 6. And they dosed with either 0.4 micrograms, 2 micrograms, or 10 micrograms. And what's really exciting is that at each of the doses, Hexapro was eliciting higher neutralizing antibody titers compared to S2P. The difference is most pronounced at the lowest dose. So at 0.4 micrograms, we're getting over a 10-fold increase. We can see a dose response for S2P going from 0.4 to 2 to 10. We don't see as much of a dose response at Hexapro, so we might be able to dial this down even lower. So not only is the protein expressing the higher levels and is more stable, it's also eliciting higher levels of neutralizing antibodies as a protein subunit vaccine. So I think that's really exciting. And then I think it's just 1 or 2 more slides just on this NDV, because Peter Placey is going to talk to you all, and I think in 2 weeks, about this NDV platform that they developed, where they introduce the active domain of the spike protein fused to the transmembrane domain and cytoplasmic tail of the Newcastle disease virus fusion protein to create this recombinant virus that still has the NDVF protein and the attachment protein hemagglutinin, but now can also put SARS-CoV-2 spikes on their surface. And it can be grown in eggs, and it produces these NDV virions that have a lot of spikes. And so they published on using the wild type spike. And then they switched to Hexapro, and they're getting about 3.5 times as many spikes on the surface of the virus. And it looks really promising from all the preclinical data that I'm sure he'll share with you. And so what we're excited about is that it combines technology from our lab and Peter Placey's lab, and it allows these countries to actually produce in-country their own vaccines using their pre-existing influenza vaccine technology, just growing it in eggs. So IVAC in Vietnam and GPO in Thailand, they've both completely made and vialed their NDV Hexapro vaccine, and Vietnam started their phase 1 March 15th, and Thailand started the week after. Brazil and Mexico are going to start, and we've also just had a lot of countries and embassies reach out. They want to make this, and yeah, we're excited. I think it's definitely going to be immunogenic, so hopefully there's just no side effects. But this could allow low and middle income countries to quickly make their own vaccines using pre-existing infrastructure and not be dependent on other countries or supply chains or waiting in line for like mRNA vaccines. All right, so to summarize everything, I hope I've shown you how structural biology has made a major impact on the development of vaccines for RSV and SARS-CoV-2. Structure-based design of pre-fusion RSV-F antigens produced some really promising RSV vaccines that are now being tested in pivotal phase 3 clinical trials. Interim results should be available in 2022. Our prior research on COVID spike structures allowed for the structure-based design of 2 stabilizing mutations that are now found in 4 or 5 COVID-19 vaccines. And Hexapro, which is a substantially improved immunogen, has entered phase 1 clinical trials as part of the NDV platform. And we're also in licensing discussions with other entities for incorporation into other vaccine platforms. In the future, we're working on development of universal coronavirus vaccines. How to define universal? Can we actually get all coronaviruses? Can we get all beta coronaviruses, lineages within beta coronaviruses? So it'll be interesting to see. And also just working on trying to identify some other interesting epitopes that can be targeted by broadly reactive antibodies, probably in S2, because that's the most conserved region of the spike. I think there's been enough work done on NTD and RBD. Really want to focus on S2. So this work's been really collaborative. Nian Chuang and Daniel led a lot of our work on SARS-CoV-2. Xinglin and Jory led a lot of it on Hexapro. Andrew and Barney, who we've been working with on all the prior studies on MERS and HK1. It's been awesome collaboration. Jennifer, Ilya, Greg, and Jason for contributing to Hexapro funding. And with that, thank you for your attention, and I'd be happy to take any questions.
Jason, that was spectacular.
Thanks. That was a lot of slides. Wasn't sure.
That's all good. So Sarada and we're a protein engineering lab, and so that was deeply satisfying, I'll tell you.
A special shout out to the collaboration and also to the speed. I mean, in a month from producing the protein, solving the structure to manuscript. Well,
no, a month from genome sequence. It was even less from, yeah. Yeah, that was sleepless nights. But it was good. I'm glad we got to do that once in my career, and we'll never do that again.
Hopefully, you
No,
know.
we'll let somebody else do it. We'll let somebody else do it next time.
Sarada, you want to start off?
No, why don't you go ahead? I'll just collect the audience questions.
Okay, fantastic. So maybe you can't tell us, but just, I don't know, above or below 50% chance that the booster shots that I'm going to get in 2 years incorporate Hexapro.
Oh, that's true. It's going to be it's going to be tricky. I don't know. I don't know. So it's interesting that it's made me rethink something. So a lot of the structure-based immunogens that we've been developing and working on, their benefit over wild type is most important and most pronounced as purified protein, the subunit-based vaccines. And you can imagine that the protein the whole time has to survive in the bioreactor, the purification, being stored in a vial. And so an NDV, though, also takes advantage of that because the virions are on the surface, and then the virions have to be inactivated, it has to survive the inactivation in a vial. As an mRNA vaccine, the differences are more subtle. They're not the tenfold that we're seeing. And so it's kind of up to the companies. I think they're deciding whether it's worth changing, whether they'd have to redo any of the clinical trials, things like that. There are some new players who don't have pre-existing vaccines that are interested, and if they're going to make one now, they might as well go with like 6 feet. So that's kind of some of the discussions we're working with.
Yeah, I mean, I bet it would. I mean, yes, it's going to be most obvious in, purified protein and like negative stain or something. But I mean, your point about dynamics was well taken. And, although it's subtle, I can imagine that you would see occasionally some profound effects. I mean, maybe not with SARS-CoV-2, but, the general notion of...
Yeah, I think it's going to depend on the protein. And like RSVF is actually more unstable than coronavirus spikes. So you might see more benefit there. We think maybe for the nucleic acid-based vaccines, it's the boost in expression that's actually more important than the stability.
Yeah.
Because a lot of this, you can make up for the differences just by giving more. So at higher doses, the differences peter out. But at lower dosing, that's when maybe a 3-fold boost in expression allows you to use 1/3 the amount of RNA, which dose sparing can be important, right? That's 3 times as many people that could be covered. But those are all business decisions.
Yeah. Okay, so other end of the spectrum, super detailed question. Do you look at proline cis-trans isomerization when you decide whether to incorporate or interpret, whether it helped or not?
No, these all appear to be trans prolines.
Okay.
We designed them. We're basically just looking at bicide angles that are compatible with the Ramachandran space of proline when trying to decide when to introduce these. If we were designing them as cis, that could be suboptimal if we needed the isomerization to occur. One of the things we're always fascinated about that we do get this boost in expression. So that's kind of our initial correlate. For RSV, we actually had antibodies. So like D25 that specifically recognized the pre-fusion conformation. So we could just test D25 binding. If it bound, we knew we were on the path. For spikes, we don't have an antibody specific for the pre-fusion conformation. So we had to kind of do everything based on just protein expression. We think the reason is that essentially for like the S2 subunit or the F protein, we have a single amino acid sequence that can encode for either pre-fusion or post-fusion folds. And that maybe while the protein's folding, there's some frustration. And by favoring it and shunting it towards pre-fusion, we're just getting a lot more protein being expressed. Mm-hmm. We haven't looked too thoroughly at that.
Yeah, okay. Another question. Can you talk a bit more about the equilibrium between the various up-down conformations? Yeah. You said you saw 1 up, 2 down in your Cryo-Em, but then when you were showing us the animation, I thought you name-checked you said 2 up, 1 down.
Yeah, there's different. So for our Cryo-Em data set, we saw essentially 1 up. David Wiesler determined the spike structure right around the same time as us, essentially using the same concept with 2 prolines. They saw a mixture of 3 down and 1 up. Some of the Cryo-ET studies, they've seen 2 up, 1 up, and 3 down. It's so we know these other conformations exist. There's pretty solid data that some of these substitutions that are occurring in these variants of concern actually increase the propensity of the RBDs to go into the up conformation to improve ACE2 binding. So early on, one of the substitutions that occurred was this D614G substitution that had a lot of publicity that essentially just took over and is now kind of the default. All the structural information and modeling suggests that favored, it eliminates a salt bridge or a hydrogen bond and then favors more RBDs in the up state. So that makes sense that maybe there's just better ACE2 accessibility. And as one would expect, though, that D614G viruses are easier neutralized because more of the RBDs are up and they're accessible both to ACE2 and to antibodies. So early on in the pandemic, when everybody was naive and there's no pre-existing immunity, D614G took over. It was a pure benefit. Now it's more of a trade-off between additional ACE2 binding, but accessibility to antibodies elicited by natural infection or vaccination. But even some of these other variants, we're seeing some of the substitutions in UK, South African variant, those are also thought possibly to alter the RBD accessibility even more. But we really need Walter Matthijs is doing some nice studies with like single molecule fret, introducing 2 fluorophores in different parts. There's always the caveat that you are introducing fluorophores, and so that might be altering. But I think those are the types of studies to really look and see how it exists at physiological temperatures on the virion.
Well, I mean, physiological temperature, but also physiological environment. I mean, I was just brainstorming maybe like deuterium labeling or something like that.
We are doing some cool collaborative work on HD exchange, and they're seeing a lot of interesting stuff that I hope that'll be published soon. So that's one possibility. That is more bulk though, rather than the single molecule. But yeah, I also don't know how to interpret like an ectodomain frozen at the air-water interface of a Cryo-Em grid. People like to measure proportions, but I have no idea if that's physiological or not. The air-water interface is so artificial.
I mean, and just I mean, whether it's NMR, Cryo-Em, X-ray crystallography, right? I mean, I do think we have to be a bit careful about like going nuts, over interpreting that sort of stuff.
64% versus 60% RBD up, and like I'm just not sure how robust that is. Or if they made a second grid, if the proportions would be totally different.
Yeah, but I mean, how would you make like testable hypotheses about like, could you come up with different vaccine candidates that tested notions of, oh, this is going to be a 2-up, 1-down candidate?
That would be really interesting. There were like 3 or 4 groups: David Wiesler's group, Prem Bhattacharya's group, John Briggs' group. They all identified the same disulfide bond, introduction of a disulfide bond that locks the RBD to the S2 and an adjacent protomer. So it's an interprotomeric disulfide bond that locks all 3 RBDs down. And it's interesting. John Briggs' group showed that it is a good immunogen actually, and does elicit high neutralizing antibody titers, which was not what I was expecting, because you're eliminating all these antibodies that could normally compete with the ACE2 binding epitope. But then it's tough because maybe it's more stable. I don't know. So still some work. But yeah, it would be interesting to test 1-up, 2-up, 3-up super unstable. You probably can't make that. 3 down and test those.
Yeah. I mean, because it seems like some sort of in situ readout is the only way to really.
Yeah, we've been doing some interesting work with Romy Amaro's group at UCSD, and they've been doing like just super impressive molecular dynamics simulations and generating testable hypotheses about the role of glycans influencing the motion of the RBD up and down. And then we've made mutants and then assessed binding. It seems to check out that some of these glycans are involved in actually promoting the up conformation by swinging into the cavity vacated when the RBD swings up.
That's amazing. That's amazing.
Yeah. So normally, because glycans are like, oh, they're just involved in like shielding from the immune system, but these actually seem to be involved in the dynamics of the RBD.
I mean, but very often they're involved in the actual interaction, not just the dynamics.
Yeah.
They're like right in the middle of the structure. So, can you circle back to, other targets? Just hit everything with proline scanning mutagenesis. So maybe like a 3.0 RSV.
Yeah, the RSV. So GSK has ours, Pfizer has theirs, Janssen. I think you could try and improve upon it. There is a certain point at which it's stable enough, right? Like going from 70-degree melting temperature to 80 probably doesn't matter. Like if you're at physiologic temperatures. So at some point there's diminishing returns.
So for RSV, can you tell us? So what's the rate limiting step there? Why is this so hard? And
it's a lot. Okay, so part of it is that the peak age of hospitalization from RSV is 2.5 months of age. So it really targets the very, very young, and premature infants and underlying conditions increase the susceptibility. So they just, those infants have really small airways. And so if they're getting pneumonia and bronchiolitis, it's really hard. So one thing is getting a vaccine in at prior to 2.5 months of age is not easy. the immune system is still relatively immature. So there's 3 different target populations: directly immunizing seronegative infants, immunizing pregnant mothers in the 3rd trimester to transiently boost neutralizing antibody titers that would protect the infant for like the first RSV season. And then the elderly population, because RSV causes a similar amount of morbidity and mortality as like influenza A. So there's 3 different target populations. And the early vaccine was this formalin inactivated RSV, and it was just disaster. So compared to the control group, it was 80% versus 5% of the infants were hospitalized after receiving the vaccine and then being naturally exposed to RSV. And so there's a lot of hesitation about vaccinating seronegative infants. And so most of what these companies are working with these pre-fusion stabilized proteins are either the pregnant women or elderly populations. And we'll see, but being able to stabilize pre-fusion that has allowed a lot of these subunit vaccines to now move forward.
What was the failure mode? Was that formaldehyde poisoning or?
So so there's a lot. There's still a lot of papers, and I don't think the field has settled on it. And it's probably just because it's really complex and that it involved anomalies in both the cellular and humoral immunity. So Barney's lab showed that the formalin inactivation triggered all the F proteins to be post-fusion F in the final vaccine product, because it was formalin activation plus heating at 37 degrees for like 2 days. And so that just triggered. So you had no pre-fusion F to elicit these really great antibodies. Other groups have shown that there was a lack of affinity maturation. And so the infants were making a lot of antibodies, but they weren't neutralizing, in part because it wasn't pre-fusion, and also maybe some TLR issues. And so you're getting a lot of binding antibodies that could form immune complexes and lead to immune complex deposition, but it wasn't actually neutralizing the virus. And then there was a whole skewing of the T cell arm into a hypersensitivity. So there's probably a lot that went wrong. And so that's what, for seronegative infants, probably the live attenuated vaccines are okay, because it's known that being infected with RSV doesn't prime you for a more severe following RSV infection. So live attenuated is okay. And then maybe something like an RNA vaccine where the infants themselves are making the antigen, it's relatively pure, that could be okay as well. But still immunizing a 1-month-old and trying to generate a robust immune response is tricky.
So do you think you would incorporate your stabilizing mutations into like the live attenuated vaccines?
So you can't do that, because then it can't grow. Because the F protein is dead then, and you need F to fuse in order to propagate the virus to high titers.
But wait,
so a live attenuated vaccine you have to be able to grow it, and F has to be able to fuse in vitro in cell culture to produce a large quantity of the attenuated virus.
But just supply the normal one in trans on a helper plasmid.
Maybe there's actually a group working on something like that, which is pretty interesting. And then there's also, maybe you could have slightly more stable but not completely dead. Marty Moore's group is working on something like that. So those are some ideas as well.
Oh, so fascinating, man. Like, I mean, your to-do list is endless. We
got some really cool stuff with human metapneumovirus. We hope to publish pretty soon of just boosted expression. Tons. It's rock solid. I think that's going to go into phase 1 clinical trials this fall. So a lot of cool stuff in the pipeline. And also now we're working on some bacteria things, trying to do something similar with like different toxins.
And so you said HA doesn't need it. Did I hear that correctly?
HA doesn't need it because it's pH sensitive. And so the conformational change is triggered by pH 5. So that's why it was the first class 1 viral fusion protein crystallized back in 1981 with Don Wiley and Scahill and Ian Wilson.
Sarada's got one.
Sure.
Yes. Jason, but before I ask my question, I just want to make sure you're okay with time, because we're 10 minutes over.
Okay, I got something in 20 minutes.
Great.
I can talk for another 5 or 10.
Too long. I have a very practical question. So regarding the immune response of Hexapro, it was promising to see the neutralization antibody levels. Are there further studies being done to evaluate the entire immune profile?
Yes, yeah, Barney's lab has been doing that, so it's kind of out of our hands, and they have a lot going on too. But they've looked at T cell responses. They've looked at the magnitude of the response against different parts, like what is causing the magnitude? Is it increased antibodies against one particular epitope, or are all responses just kind of amplified? And they're trying to tease that out now, and hopefully they can publish on that soon.
Oh,
We've also done work with Moderna, so we do have data comparing mRNA 1273 versus Moderna-produced mRNA encoding Hexapro. That's pretty interesting. It's just taken a while to get it published, so I can't really talk about it.
Cool. We look forward to it. Then the other question is, apart from, I mean, of course, these are designer epitopes for future vaccine designs, but what other applications do you think we could use? I mean, I would imagine this could be a good target for, those in-home testing kits, because those, they probably would be this. I mean, this.
Yeah. We have licensed. UT has licensed Hexapro to a lot of different entities for different reasons. And a large one are for companies that are performing diagnostics, serological studies, CROs that need to test vaccine responses with like ELISAs and other things to spike. This is like the easiest one to make and most stable. So we're seeing a lot of that.
Wow, that's exciting.
Jason, thanks so much for your time and thanks.
Yeah, no, I'm going to tune in when Peter Placey talks, because I haven't seen all the Hexapro data they have. I've seen some of it, but kind of curious to see. I know they're writing that up, but that should be cool to see the data.
That's really exciting.
Great. Well, thanks for the invitation. It was great speaking with you guys, and look forward to seeing you for Peter Placey's talk.
Yeah, and thanks for reminding everyone of the power of protein engineering.
All right. Cool. Take care.
Thank you.
All right. See you, Jason.
Bye.
Matt, do you have the slide for? There we are. Tune in next week. Hear Tyler Starr from the lab of Jesse Bloom give us a fantastic update to their saturation mutagenesis of spike protein and neutralizing antibodies. So stay safe and see you next week. Bye.